is Michal Francel, and I'm here to present my work on segmentation of whole slide images with context-aware vision transformers. It is important to mention that it is still a work in progress. Histological analysis is a crucial part of breast cancer diagnosis, and prepared tissue is placed on a glass slide and stained, and as a result, and it is scanned, and as a result, we obtain gigapixel images that are demanding to process and are hierarchical in their nature, and as such are also hard to analyze, and because of that, there is a low quantity of annotated pixel level annotations. Our objectives in this work are threefold. It is to firstly apply transformer-based models which obtain state-of-the-art performance across other computer vision tasks to domain of breast cancer pathology. Then secondly, it is to enhance these models in such a way that would allow for the usage of multiple contextual magnifications to provide context and further improve model. And lastly, we aim to employ semi-supervised methods to address the issue of the lack of pixel level annotations. Moving on to the related work, in this systematic study, authors studied the effects of usage of multiple levels of magnification for context. They achieved the best results while using late feature fusion from lowest to the highest level of magnification. And this highlights the importance of incorporating context from multiple magnifications to the model in order to enhance the predictive capabilities of the model. Next, there is a hierarchical transformer for histopathological images that aggregates features from bottom up, from highest magnification to the lowest. And its goal is to create a single representation for entire whole slide image. Interestingly, they achieved this by utilizing Dino pretraining framework and pre-trained vision transformer. And this demonstrates the potential of applying vision transformers and self-supervised learning frameworks like Dino to the modeling of histopathological images. And moving on to our proposed solution, we worked with two datasets within our experiments. First is Bragg's dataset that contains 547 whole slide images that are annotated into six types of lesions. We use these images within our self-supervised experiments without labels. And the second dataset is Tiger dataset, from which we use BCSS part that contains 151 selected regions that are annotated with segmentation masks. And you can see there are numbers of those regions within those classes in a table below. We have chosen a semi-supervised method consisting of unsupervised pre-training and supervised fine-tuning. We process entire whole slide images at the input, and we process them using a patch generator. For the self-supervised method, we selected iBot, which operates on the principle of modeling masked images. and knowledge distillation, and we analyze its properties in greater detail together with learned features. And simultaneously, we explore the possibilities of using contextual information within the supervised fine-tuning part of our work. First, the generator generates a tissue mask through thresholding and morphological operations to avoid training on empty regions of the slide. And second step is creating patches, with dimensions of which are determined by pixel size of pixel size in microns, and then we process contextual patches from lower levels of magnification and create a stack. As for training, we also use, as for supervised training, we also use augmentations, horizontal vertical flips, random rotations, and contrast-limited adaptive histogram equalization. As a loss function, we use dice cross-entropy loss. And during our supervised stage of training, we used all 7 annotated classes for segmentation. First, we experimented with using just a single scale without contextual information. We compared convolutional and swing-based units, which performed similarly. Then we experimented with more advanced segformer and APRNet networks with transformer-based backbones that performed significantly better, with APRNet performing with dice score of 56.05. And so we chose this APRNet for next experiments. This is how APRNet works. It contains pyramid pooling module, which takes last features from last stage of encoder, and FPN fuse model, which upsamples and fuses together features from other stages and combines them to create segmentation mask. Then after initial experiments, we moved to experiments with multiple scales used for to provide contextual information. We experimented with early fusion, intermediate fusion, and late fusion of features, both with and without cross-attention blocks as a mechanism for transferring information. And we found that late fusion without cross-attention works the best, achieving dice score of 62.25, which is about 6% better than without using contextual information. We used free swing encoders and combined features with linear layers and concatenation. And this, on the right side, you can see evaluation per class. We can see that performance was a little bit worse within class other, but other is also a combination of several other classes that do not necessarily look the same. And this is the prediction. This is an example prediction of the segmentation network utilizing 3 magnifications. We can see the other class is misclassified. Then within our experiments, we moved to self-supervised analysis of self-supervised pretraining. We attempt to improve model with iBot pretraining framework, which uses knowledge distillation with student and teacher, and we try to analyze and pre-train a vision transformer, first with and then a swing transformer. We trained on all 3 magnifications, and we found that within highest magnifications, model started to focus on cells and stroma, and could differentiate also between fatty tissue, which is displayed here. Then within lower magnification levels, model performed worse. It started to focus only on darker and lighter spots within images, and as such we evaluated that it is better used for higher detailed images from higher magnifications. Then this is TESNI projection from Brax dataset. We can see that model learned to differentiate between various types of regions from images. And after evaluating on BCS dataset, labeled dataset, we found that it learned to differentiate also between classes that are contained within the dataset. And after fine-tuning, we discovered that it significantly improved with in situ tumor, healthy glands, and also within our other classes, mostly within classes that are visually different from each other. So as for conclusion, we had firstly experimented with single-scale architectures, comparing transformer and convolutional networks. Then we moved to experiments with multiple scales for to provide contextual information for the model. And afterwards, we tried to improve the model further by self-supervised pre-training with masked image modeling with unlabeled data. Thank you for your attention, and now this is time for discussion. So if you have any questions.
For like 7 minutes for questions plus minus. So let's start. Okay, Victor.
The pre-training is done only with encoder. So when we look at this, we pre-train only only the encoder part.
Multiple scales experiment and the third experiment, the knowledge exploration, was also the same architecture, or you used the visual transformer?
For-
Because it was the same architecture in this multiple scale experiment, or also the third experiment?
With third experiments, we trained or pre-trained with only a single scale, one scale at a time, and it is up for future work to maybe combine the pre-training process so that all scales would train at the same time. Our first experiments were pre-training VIT, or vision transformer, because it is much easier to visualize attentions. So we can visualize what the model focuses on, basically.
So your goal is to use the architecture from your second experiments also with this kind of techniques of knowledge distillation? Or like I
Yes.
how they are connected, the second and third experiments, so your goal is to continue the system. How do you want to connect them? Is there a plan to connect the second and third experiment?
Plan is to possible plan is to further modify the pre-training stage so that all three encoders from those three branches that I mentioned are pre-trained simultaneously and then used to initialize weights for this architecture.
Okay, so you want to do the pre-training from this third experiment and apply to this architecture in the second experiment later?
Yes.
Okay. Are there any other questions from the audience? Okay, I have maybe one. So are you also dealing with different colorants in your samples, in your database, or it's like all the colors are the same for all samples? Or there are none?
I didn't understand. Color?
Okay, if the data on the input that you have, that if they are also stained by some colorants.
Yeah, they are stained by hematoxylin eosin, and to normalize, we used also Matsenko normalization within the fine-tuning process, so that we normalize BCSS dataset, fine-tuning dataset, relative to the pre-training dataset, Brax dataset.
Okay, so probably there is no problem with these differences, and the normalization works fine for you. Or can it be improved? Like I think that this Matsenko normalization, I'm not familiar with it, but does it work fine? Like, is it something which is like state of the art and working very well, or are you having some problems with it and can maybe be improved?
It worked very well, I think.
Okay, thank you. So are there any other questions maybe from the audience? Okay, Victor?
Overall, there were 151 images that are or regions of interest that are annotated with segmentation masks. From them, around 30,000 patches were created of size 224 times 224, and 151 regions were divided into training and validation data 90% of rows were within the training data and 10% within validation data.